Welcome to Life in Biology. I'm Dr. Joel Graff, and today's topic is about virus genomes and the variety of different ways viruses go about carrying on their genetic information. Okay, so what better way to start a discussion on genomes than with the central dogma of biology? So does this make sense? I want to talk for a second about sense and antisense strands. So central dogma of biology, we have DNA, and I've drawn it as a double-stranded molecule here. That's the way I draw DNA molecules, is basically a ladder on their side. Then we have DNA gets transcribed to RNA. RNA is a single-stranded molecule, and so you only see the top or purple strand here. And then RNA gets translated to protein, and I'm not much of a drawer, so this is my example of what a protein looks like. Okay, so let's take a look at these drawings a little more closely. I tried to do some color coding, where in my pictures I'm going to have what's known as the sense strand of either DNA or RNA be labeled in purple. And there are multiple terms that could be used for the sense strand. Another example is you might call that the positive strand, and we'll get into that a lot more in this video. And then if you have a sense strand, the strand of DNA opposite of it is going to be called the antisense strand. And I put a plus sign in there, and that is wrong. It should be a minus sign. So that's a minus sign if you can't see that. The antisense strand is also known as the template strand, and the sense strand is the non-template strand. The reason that this is the sense strand is that when transcription occurs, the polymerase will actually read that bottom strand when it's making the RNA that's of the top strand there. So this is the sense strand, and this is the nonsense strand. So lots of different terminology here, but we'll just mostly stick with positive and negative strand from now on. But we'll just know that that positive strand is the strand that can be read and made into a protein by the ribosome. All right, all viral roads lead to positive single-strand RNA, or I could say mRNA, but in this case we'll say positive single-strand RNA. Now, a guy named David Baltimore came up with a system of categorizing viruses based on their genome type, and if you are told the type of genome that a virus has, you automatically, if you're a virologist, think back to the Baltimore System and understand several features of that virus's life cycle. So, starting with group 1, the Baltimore System uses Roman numerals, so group 1 is double-stranded DNA, and double-stranded DNA can be transcribed to RNA, which can be translated to protein, and that sounds like the central dogma because it is. Now, all living things, if you assume that viruses are not living, follow the central dogma of biology, and a group of viruses also follow the central dogma of biology in their genetic flow. So that's group 1. That's the one that most people familiar with biology should be most familiar with because it is the central dogma. Now, group 2 is when you start with single-stranded DNA, and in this case it's a positive single-strand DNA, and then you have a polymerase make the other strand, the minus strand, so that you get to double-stranded DNA. So this is the genome. This is an intermediate in that virus life cycle. The double-stranded DNA can then be transcribed to positive single-strand RNA, which can then be made to protein. So we've got through groups 1 and 2. Rotating over here, we get to group 3, which is the double-stranded RNAs. And double-stranded RNA, if the polymerase reads the red strand, it can then make a copy of the purple strand, the sense strand, and then you end up over at protein. So notice with group 3 viruses and a few more that we're going to mention, you don't have any DNA involved. DNA is completely taken out of the picture. We just go DNA or double-stranded RNA to single-stranded RNA to protein. That was group 3 viruses. Now we're going to go on to group 4 viruses. And these viruses are interesting because when they release their genome, when they enter a cell, it's automatically, or it can be automatically read as an mRNA, and it can be automatically translated to protein. So we've skipped out any previous steps. We just jump in at the key point of positive single-strand RNA. Moving on, after group 4, we have group 5. In this case, it's single-strand RNA. When these viruses enter the cell, their single-stranded RNA can't be read as protein, and instead, an RNA-dependent RNA polymerase needs to read the red line and make a purple version of it so that you have a double-stranded RNA intermediate. Those top strands can be used then as the positive single-stranded RNA or as the mRNA, and then you can be made into protein. Again, we don't touch DNA in that case. Now, groups 6 and 7 are a little contentious because it used to be that groups 6 and 7 were two separate things, but there is some push to combine groups 6 and 7 because they both use the reverse transcriptase enzyme. Reverse transcriptase, remember, is the act of going from RNA back to DNA, so it's the reverse of transcription. And so if you start with a single-stranded RNA that's negative sense, notice we've got a single-stranded RNA here, and group 5 was also single-stranded RNA, but it's going to act differently. Rather than up here, it was an RNA-dependent RNA polymerase that brought you back to a double-stranded RNA. It's going to be an RNA-dependent DNA polymerase, which will give you a DNA purple strand, RNA red strand hybrid. With that DNA-RNA hybrid, then I forgot an arrow here. If I had an arrow, I would have an arrow drawing up to double-stranded DNA from that DNA-RNA hybrid. Apparently this was a little bit too complex of a slide for me to work through. Then you're at double-stranded DNA, then you go to single-stranded positive sense RNA, and then you go to protein. Over here is a partially double-stranded RNA molecule. It has a weird circular-ish genome where the two strands of DNA work together to basically make more or less a single-strand circle, but it flips strands. It's complex. We won't get into all the complexity of that right now, but if you were to have an RNA or a reverse transcriptase work on this, it could use one of those strands as a primer, and you can get to a DNA-RNA hybrid, and then you can go back to double-stranded DNA with the arrow that is missing here, and then you go to single-stranded positive RNA onto protein. Okay, so that's showing you how much variety there is in RNA genomes. But again, the main point that I want to get by on this is that everything has to get to a single-stranded RNA positive sense, because the host cell that the virus is infecting has the translation machinery that allows you to go from a single-stranded positive sense RNA and read it like an mRNA and make a protein. So viruses could be considered parasites of the host cell's translation machinery. We're getting some really giant viruses discovered these days, and they actually have some of that translation machinery in. So the line between being a complete parasite of host genome versus having some involvement in it from the virus is beginning to get blurred. But again, mostly you can think of viruses as parasites of the machinery that goes from this step to this step. And as long as I was mentioning all those groups of viruses, I figured I'd make a quick note of what are some of the viruses that fall into these different groups. So we've got our viral groupies here, and I've listed them 1 through 7. Now this is not an exhaustive list. Group 1 viruses, for example, herpes viruses are an example of those, but so are adenoviruses, and so are pox viruses. So we're not talking about everything here. And then we have group 2, parvoviruses is an example. Group 3 are reoviruses. I did my PhD work studying one of the reoviruses, which is called rotavirus. And recently I've been hunting for some cultivirus out in the mountains around western Montana, which is a virus that's carried by a tick, but those are all examples of reoviruses. Orthomyxoviruses, you might not have heard of that word before, but you probably have heard of influenza virus. Influenza virus is an example of an orthomyxovirus, and that's influenza causes the flu. So flu is just a shortened version of influenza virus. Okay, then we have some lentiviruses here in group 6. Maybe the most famous of those would be HIV-1 and HIV-2. And then group 7 are the hepadinaviruses, and those are that weird circular DNA genome. Okay, so there's just an example. I do want to point out that there are tons and tons of positive single-stranded RNA and negative single-stranded RNA viruses that infect humans. So again, just remember, this is not an exhaustive list of the different virus groups. Okay. Well, now you know about virus groups and the way that we can categorize viruses. Have a good day. Like and subscribe, or not.